Greetings and thank you to the 12 of you who are watching this talk online. I wanted to talk about a highly collaborative effort that we started right before the summer between the UH Manoa Group, Oregon State University, and Zeiss. This talk is focused on some preliminary results of this effort and how they might be used to better understand clustering of minerals in magmas. Before we talk about a relatively new analytical imaging technique we used, I wanted to introduce the guiding underlying question behind this study. How are crystal clusters formed? And notice we try to use the term clusters here to avoid making any inferences about grouping mechanism. So aggregate or glomerocrystal, for example, implies separate objects that were later brought together. What is clear from decades of studies of crystal cargoes in volcanic rocks is that clusters are widespread in magmas. And they're of fundamental importance in understanding magmatic systems because they may well represent the building blocks of igneous rocks. And I'm showing here a collection of clusters consisting of only one mineral type, from olivine at the bottom to quartz in the middle, spinel, or plagioclase. And there are three commonly proposed mechanisms in the literature for cluster formation: growth, accumulation or settling, and synusis. Over the next couple slides, I will try to summarize some of the basic features of each of those models. So accumulation or mineral settling is probably the oldest of the three, where gravity and density differences between mineral phases and their host melt drive the settling of minerals towards the bottom of a magma reservoir. And some of the predictions of the model are that crystals can be randomly oriented within a cluster, sort of like this nice example from Camejo Harris et al., and that each settling crystal may have its own separate zoning history. And this settling mechanism has been demonstrated time and time again experimentally by poor petrologists who would rather have nicely distributed crystals within their charges but end up with many mush piles like this one at the bottom of their charges. Synusis is a second mechanism that was proposed by Vance in 1969, where crystals aggregate when they are still swimming together in melt-rich magma after making initial contact. And the predictions made by this model are that, for one, crystal faces would tend to align against each other after initial contact in order to minimize surface energy. And so this would explain the presence of crystal clusters that often seem to have grains with parallel or non-random orientations, like the olivine at the bottom here, or the plagioclase in the Vance paper. Like with aggregation by settling, each separate grain may have its own zoning history, as illustrated either in, again, this zoned plagioclase crystal, or in the iron-magnesium zoning patterns within this profile collected by Schwindiger and Anderson. And those have apparent zoning and cores that suggest these were initially separate crystals. The third clustering mechanism was one that was initially mentioned in an older paper by Drever and Johnson, where they talked about parallel growth of olivine and budding. Doughty later formalized the notion that cluster appearance could result from these three variants of growth in his 1980 paper. One, skeletal growth and branching can create series of budding grains. Two, nucleation and growth of a new grain around another grain is a second mechanism to produce clusters. And then three, this is the well-known growth of two or more grains along twinning directions. So growth can explain why clusters have parallel orientations or orientations that are consistent with twinning laws. But other studies have also shown that growth can lead to radially distributed crystals. And these are examples of radial growth of plagioclase within experiments at the top and within these natural samples at the bottom. Importantly, and lastly, it's been suggested before that clusters related to each other by growth should show similar zoning histories. But in reality, zoning can be sequential as new buds or twins are growing one after the other. So we'll talk a little more about that later. Okay, so on to our topic du jour, characterizing clusters in 3D using X-ray tomography. Most of us are familiar with absorption contrast X-ray tomography, or ACT, where a sample is scanned along various directions with X-rays, and those X-rays are attenuated depending on the density of the material that's crossed. So essentially, ACT allows us to resolve materials with varying density in 3 dimensions. Well, Zeiss, just a few months ago, released a new instrument called the X-Radia Crystal CT, shown on the left here, where X-rays can be used for both their capacity to resolve differences in densities, but also resolve differences in grain orientations through diffraction contrast tomography, or DCT. And the geometry of the setup is shown here, where a polychromatic X-ray source goes through an aperture and hits a sample that rotates, and the outcoming signal is partly blocked by a beam stop to avoid damaging a sensitive detector that is sensitive to the main powerful transmitted X-rays. Instead, the weaker diffraction signal is recorded as spots around the screen. I can't spend more time on the method, unfortunately, but you can read more about the technique in these 2 papers by Oderschede and Pankhurst et al. This is what one DCT image from one particular crystal region might look like. And when we can, from these diffraction spots, recover the orientation that best match these spots through the use of available libraries and reconstruction algorithms in this software called Grain Mapper 3D. And this is the resulting 3D rendering. And this technique can resolve diffraction signals coming from 2 objects that are down to 20 microns apart, and the angular resolution is 0.1 degrees, meaning that 2 grains with misorientations greater than 0.1 degrees can be distinguished from each other. So for the 8 of you left watching by this time in the talk, we ran 2 first tests with crystal CT on olivine clusters from a kilowatt eruption. And I'll first show you a video of the absorption contrast scan, or rather a reconstructed 3D rendering, after we segmented out or separated the different phases like olivine, chrome spinel in yellow, melt inclusions in orange, or bubbles or fluid inclusions in blue. So as the volume rotates, you notice that some of the subgrains of this kilowatt cluster seem fairly parallel, but others not. And as we kind of make it more transparent, you'll notice that the number of spinel is enormous in this particular olivine, as well as the numerous melt inclusions and bubbles. But this is for another time and another talk. On the right side is a 2D DCT slice through exact same crystal that is then rendered in 3D at the bottom with arbitrary colors that show the grains that were identified as having different orientations. Okay, so on to our main results, and sorry for the pretty busy figure here, but the plot shows a parameter called the misorientation angle on the x-axis, which one can perceive as the difference in orientation between the crystallographic axes of 2 grains. The y-axis displays the number of touching grain pairs that have this given misorientation angle for the 2 clusters that we analyze with DCT. Behind these histograms, I'm showing green bars that would correspond to things like parallel growth or near 0 degree misorientation, and those that correspond with known twinning angles in olivine. So essentially green things kind of consistent with growth-related clustering. I also placed blue bars that might correspond to orientation of grains that are attached via common face-to-face angles. I did forget one for parallel growth, which should also be next to the 0 here. So this would be, the blue ones would be more consistent with aggregation and or sinuses. To a first order, it seems like the distribution of grain misorientations into the 2 clusters is slightly closer, at least the modes, to the green bars, but not always. You can see that the blue ones are also close by. In essence, so purely on the basis of grain orientations, we can't really confidently say that growth or sinuses is the only responsible mechanism to form clusters. So what about zoning patterns? As we mentioned earlier, the presence of apparently different zoning histories within clusters has often been described as a symptom of aggregation because it marks distinct crystal histories that later came together. So this is another set of profiles from a more recent paper by Wieser et al. And we wanted to test whether this assumption held. So we took our scan 3D olivine cluster model to carry out numerical zoning models. The starting point is that we give a fully homogeneous starting composition to our cluster. We then developed iron magnesium zoning via diffusion and cut our 3D model randomly to evaluate whether sectioning effects might be important in what we ultimately read and interpret from the 2D thin section record. And this is one example slice. And these are other examples where with their corresponding iron magnesium concentration profiles. And the problem here is that zoning can progress to different extents inside grains depending on their size, so that their cores can possess different compositions. And then you have sectioning effects on top of this that exacerbate by cutting grains or clusters off center. Intersection effects can really give this false sense of grain and grain zoning separation. So these zoning profiles clearly show the exact same symptoms previously identified as implying different crystal histories when, in fact, they all share the same petrologic history within our simulation. So I would argue that 2D zoning in thin section does not provide a means to carefully discriminate between growth, aggregation, or synosis. So what can we do to actually solve the question of cluster origins, particularly when we have clusters with preferred grain orientations? In our mind, the only way to do this is to gain a three-dimensional view of natural zoning. We need to determine how zoning varies between and at the contact between grains using elements that best record initial crystal growth. And for olivine, that would be phosphorus. So at the moment, X-ray tomography does not allow us to resolve zoning in things like phosphorus, and hence we've decided to take the brute force approach of making serial sections repolish, remap with the microprobe within one of our olivine clusters to try and see if we can spot some symptoms of growth or synosis. And these are idealized 2D phosphorus zoning sections, and they give an idea of what sort of zoning results we might anticipate for one or the other close clustering mechanism. If growth is responsible, we might see things like surface nucleation as a new core on an existing seed and asymmetric growth around it. We might see branching and formation of secondary buds. We might see cores and zoning related to twinning. If, on the other hand, aggregation or synosis processes dominate, we would expect to see phosphorus zones that mark well as separation and this face-to-face preferred contact. So here, the five of you left in this presentation might say, well, this is a bit foolish, and how many serial sections will you need to make, and how much mapping time will this take on the microprobe? And you would be right to say this because we are far from completing the exercise. In fact, so far, we have 12 serial sections of our Kilauea cluster, and while we are starting to see some interesting zoning relating to growth, like here in this zoomed image of the 12th one, we have yet to reach the interesting part of the cluster where the grains are really adjoining. So I invite you to Season 1, Episode 2 of this exercise, perhaps at Goldschmidt next year in Honolulu. Okay, so closing thoughts. All models have pros and cons. Aggregation settling is easily understood, but does not always explain preferentially oriented crystals. Growth is too often dismissed because of zoning differences between grains, but sectioning effects matter greatly. Synosis is often invoked, but the exact force required to move grains once they touch is unknown or experimentally has never been demonstrated. So essentially, we need a lot more data, and we need to understand zoning in the third dimension. And we can use not just phosphorus and olivine, but for example, anorthite in plagioclase, barium in sanidine, titanium in quartz, chromium in CPX. All of those slow diffusers will mark the initial growth history well. So a blinking red light on the podium tells me I'm going to get kicked off stage. So for the single person that finished this virtual presentation, thank you for your bravery. Please email me or ask questions on Monday from 8:50 to 8:55 a.m. New Orleans time. In the meantime, I hope to have convinced you that solving the clustering problem should be as easy as playing Ode to Joy on the recorder.